So this is used to compensate for any variability of the experimental conditions, because you really want to make sure that what you see is not a one-time event. It's really, how do you say, the effect of either your bioactive chemical or whatever you are using in there. Then medium composition. So the cell culture medium is further vital, providing key nutrition and also influencing the cell behavior. And choosing the optimal medium ensures an accurate representation of the biological process. So if you're studying any bioactive components, your wound healing assay, it is also necessary to establish the ideal concentration of this. And finding these concentrations is best achieved with a dose-response curve. This helps to find the balance between the desired cellular response that you're looking for, and on the same time also avoiding any cytotoxicity. Furthermore, we would also always suggest you, when working with these components, to look into existing literature and also recommendations from the manufacturer. Then next, imaging. So imaging is really playing a huge role in wound healing assays, as it allows for the detailed observation and analysis of cell migration in real time. The cells should be monitored immediately after the gap formation, and usually it's then sufficient to monitor the cells more or less 24 hours after, or even less depending on the setup. And during imaging, a stable and physiological environment should be maintained to ensure a natural cell behavior. So if you perform the assay as an endpoint assay, it's possible to go back and forth between the incubator. But if you're looking to do a live cell imaging setup, it can be necessary to have a cage surrounding the microscope or a stage-top incubator. And the use of such stage-top incubator ensures that the cells are maintained in an optimal condition, while facilitating this continuous monitoring of the same gap position throughout the entire experiment. And the advantage looking at living cells with a live cell imaging setup is that we get real-time analysis and real-time data, and therefore also an increased information output. The cells here are intact and they are functioning and interacting with each other. And with these live cell imaging, you get real footage of how the cells migrate. So yeah, as stated, how they interact and function. You see an example here. So these are 2 movies and from wound healing experiments. And based on these 2 still pictures, we would have never known that these cells have different migration styles. That the fibroblasts here to the left, they need cell-cell contact to move forward, so they do so in a very line-based manner. I will just switch the laser off, so you see it here. They really need this contact, and then they move forward as a line. While if we instead look at the keratinocytes over here, these cells are not needing any cell-cell contact, so they're just jumping forward, then closing the gap very independently. But yeah, as kind of hinted before, there is a challenge with these live cell imaging setups, because we would need the cells to stay healthy on the microscope for longer times, and therefore something like a stage top or a cage is needed. And to support live cell imaging, EBD offers such stage-top setups. So this is a small incubator, it's very handy, and it can help control both the temperature, humidity, and gases to ensure that our cells are viable for the entire duration of the experiment. And if interested, we actually offer free demos of the system, so you can test it out on your own inverted microscope. But we actually also, as a new thing, have a course on the topic, so a course specifically on live cell imaging and the stage top, and it's actually already next week if you're interested in listening. Okay, any questions so far?
We had one question, maybe that's interesting for everybody. I already answered it shortly. We have a free sample program, and I think you will probably at the end mention it.
Yeah, also how to get the samples, and how come a slide.
How to get it. It is, so everything you will see, which is an EBD product, is available as a free sample, so you can try it at home or in the lab, not at home.
Yeah, I guess it's more relevant in the lab, yes.
Yeah, it's more relevant, but in the home lab.
Yeah, so all labware is part of this free sample program, and all instruments presented, we have free demos if you want to test that as well in the lab.
Yeah.
Okay, anything further?
I think I could cover all the rest.
Okay.
By text, so we can...
let us know if there's anything that is relevant for everybody, because I think we are very nice in time, so there is time for it. Good.
Okay, yeah, then I will maybe save something for...
Sounds good. Discussion.
For the discussion, yes.
Okay, the last point to consider before initiating your wound healing assay is also the gap creation method. So the method of creating a cell-free gap is a crucial factor in wound healing assays, directly influencing the cell behavior, but also the reproducibility of your experimental data. And all different methods available have, of course, a list of pros and cons, and it's important that you choose the one that is correct for you. So what are you looking to investigate? But we can now go through a few of these different assay types. And we can start here at the top, so with mechanical wounding assays. And this is the most common method for studying collective cell migration. We already have somebody commenting in the chat that they did this. This is often chosen because it's very simple and cost-effective. In a mechanical wounding assay, cells are disrupted physically and the collective cell migration process is then observed. And mechanical wounding can be categorized into mechanical scratching and stamping assays. And in a typical mechanical scratching assay, cells are allowed to grow until they reach confluency, and a mechanical wound is then created by physically scraping the cell monolayer. And the scraping can be performed using, for instance, a pipette tip, needle, razor, or something similar if you want to create a single scratch. Or you can also use a plastic comb for creating multiple scratches simultaneously. And to standardize the process and also increase your throughput, there are also people that use scratching assays where they implement a robotic system. However, at least what we see for most of our users is that they do this manually by hand instead. And here's an example of a scratch assay performed with a pipette tip. So the time-lapse images and also the gap closure measurement detects as this gap is then slowly closing again afterwards. Yeah. And this method, as stated, is quite straightforward, but since the scratching is often performed manually, it can lead to some challenges, mainly influencing the consistency and also reproducibility due to the differences in the force that is applied upon creating the gap. On the same time, it's also very time consuming, so having to scratch down every single monolayer precisely every time. On the positive side, here we are really disrupting a cell monolayer, so we create surroundings with injured and dead cells which help mimic the presence of a wound. Next up is chemical methods. So the cell monolayer can also be wounded using chemical methods. So for instance, sodium hydroxide has been used in several applications to lyse cells and create wound models chemically. And in this approach, a small droplet of sodium hydroxide is pipetted onto the cell monolayer to selectively remove cells in contact with the droplet. And therefore, the size of the wound is also controlled by the volume of the chemical that is applied. And another example is shown here of a chemical wounding. So this example was created in the EBD lab. Here fibroblasts were cultured on a photoresponsive substrate, and upon illumination with blue light, it was releasing singlet oxygens. And these radicals then induced apoptosis in the cells that were growing on the illuminated area. And this method really makes it possible to create a defined lesion in a cell confluent layer, and also leaving the dead cells behind in the wound. So like the chemical method, electrical wounding methods, so based on this electrical cell substrate impedance sensing technique, can also create wounds with different geometries. The electrical wound healing system typically is composed of a working electrode for cell adhesion and then a counter electrode. And then by applying a relatively large voltage between the electrodes, the cells on the electrode can then be electropermeabilized to generate a model wound in the monolayer. And because of the insulating properties of the cell plasma and membrane, the cell movement can then be characterized by the measurement of the electric impedance of this monolayer afterwards. And here's an example of such assay. It's performed with HULEX. The assay was conducted again in the EBD lab using the ESIS instrument from Applied Biophysics. And on the images to the right, you see an overview of the electrical induced wound at different time points. And this was then further recorded to the left as this impedance over the cell layer during the entire experiment. Yes. And wounding of the cell monolayer can also be performed as an optical method, so using a laser. And in this method, the cell is in this predefined wound regions is eliminated by a laser ablation. And the motion of the laser pulse and the microscopy observations can often be computer controlled. And such automatic methods are therefore more suitable if you're doing multiple assays with a very high efficiency. Other advantages using such laser methods is also the ability to create repeated wounds and wounds with arbitrary shapes. And lastly, we have the physical barrier assays. So a physical barrier is another method for creating this wound healing assay. So here we are creating a cell-free region for collective cell migration. So instead of removing cells in the monolayer, we have a physical barrier that is applied before cell seeding to block cell attachment in certain regions. And the removal of this barrier then initiates the cell migration process. And the repeatability and also standardization of the physical barrier assay is compared to the classical wound healing with a scratch assay, more, standardized and possible to repeat. This was actually proven by Melanie Boes at the University of Freiburg. So she compared the gap closure over time between using the EBD culture insert, which is a physical barrier, and then a classical scratch assay. And in these two graphs, it's very clear to see that using the physical barrier assay as these culture inserts, it's possible to decrease the variation in the gap size and thereby have less background noise in your experimental setup. Okay, and the use of a physical barrier also minimizes injury to the cells and keep the cell-free region intact from extracellular matrix deposition. And the smoothness of the surface after removal of the barrier, so with no residues left on the surface, makes it relevant for tons of other migration studies than just wound healing. If you have